right, good afternoon, everyone. I appreciate all of you bearing with me while I'm getting over a bit of illness here, but I did want to get you guys a lecture just so that we can move into myocardial infarction, as we are running a little bit behind, but it's okay. So when we left off in class, we were talking about this beta-1 adrenergic G-coupled protein receptor. And we were talking about these subunits that are located here, the alpha, the beta, and the gamma. And there's a lot of research out there that indicates that all of these subunits do move independently. Some of them indicate that the beta gamma subunit doesn't really move and signal. Others have found that the alpha subunit moves independently with the GTP, as we talked about here, right? That alpha subunit is activated because it's carrying that high energy guanine triphosphate, right, which is activating it. So we did talk about this signaling pathway with the A or the alpha stimulatory pathway, which means this is going to stimulate contractions in the heart, and it's going to do so through the mechanism of increased sympathetic overflow. And we know that that is going to activate the alpha subunit, as we talked about in class. And that alpha subunit, in order to be active, it's going to exchange that GDP for this GTP, and that's what's going to allow it to mobilize and translocate. Well, the first target that the alpha subunit is going to interact with is this adenylene cyclase. And please keep in mind that even though this is kind of heavy cellular stuff, if we were to look at the amino acid interactions between these two proteins, right, we have the alpha subunit and we have the adenylene cyclase, which is an enzyme. Just how they interact and the regulation between these two proteins is very, very complicated as well. So we could zoom in more and look at this interaction between these two proteins, which we're not going to do yet, but maybe further down the road we will do. We know that this adenylene cyclase is an enzyme, and once the alpha subunit interacts with it, it is going to take ATP, which is circulating in the cell, right? And we talked about why that is so important to have lots of mitochondria in the cardiomyocytes in chronic heart failure. That mitochondria will start to perish, which means the ATP will start to be lower, which means the ability to activate cyclic AMP will be lower. And we did talk in class that cyclic AMP is also considered a secondary messenger. And this secondary messenger is going to amplify this signal, right? It's going to amplify this initial signal that is coming from outside of the cell. So in amplifying it, we know that cyclic AMP is capable of binding to, or we could just say interacting with the SA node, right? That's going to increase firing of the SA node. We also know that it is going to phosphorylate or turn on. Remember, I told you we have like a light switch. If I have on and I have off, well, if we want to turn that switch up, generally we have to put a phosphate onto a protein, and that turns it on. And again, this is a very simple explanation, but you get the gist of it. I'm going to take off some of this writing before it gets a little too, before I forget what is old and what is new. So I'm just going to kind of remove that. And we know that once pKa, which is this protein kinase A, so we know it's a kinase, right? Which means this is an enzyme, which means this is going to cause a reaction. We know that pKa is capable of doing roughly three things. It is going to phosphorylate and activate L-type calcium channels, which means calcium is going to flood into the cardiomyocyte. That's really important because when we have these contractile tissues in the cardiomyocytes, such as myosin and actin, they are going to need calcium to regulate their contraction. We know that the pKa is also going to phosphorylate the Circa pump, and we know that this is on the sarcoplasmic reticulum. We know that this is inside of the cell, and I'm just going to say that this is the circa. I'll just put an S. And we know that the circa pump is going to regulate calcium out and then calcium back into the sarcoplasmic reticulum. And that's really important because we need time of heart relaxation where calcium is not flooding the cell, right? There needs to be times where there is a lower concentration outside of the sarcoplasmic reticulum than there is on the inside of the sarcoplasmic reticulum, and that's going to give that myosin and actin a chance to kind of rest, right? So when we're resting, we want high calcium concentrations inside the sarcoplasmic reticulum, and we want low calcium concentrations outside of the sarcoplasmic reticulum. That allows myosin and actin to kind of disengage, cause relaxation, allow the heart to start filling the atriums of the ventricles wherever we're filling. But if this sequence is disturbed because we have overactivation of pKa, well, that's going to directly feed back and impact our ability to let proper filling of the ventricles or the atrium to occur, because so much calcium is causing an increase in contraction, which is causing a decrease in diastole, which is causing a decrease in volume filling within the heart, right? So you can see how all these little changes are associated with complications of contraction in the heart, right? And it's all, thank you so much, epinephrine, norepinephrine. We appreciate you being on constantly, right? So let me clean this up. And then, as we mentioned in class, the last thing that's kind of occurring is that the pKa enzyme is also relaxing the troponin protein that is surrounding myosin and actin, right? So this stimulatory pathway is stimulatory because it is supporting contraction, right? I'm just saying plus sign, saying that, hey, it's creating an environment in the cell to enhance contraction, right? And the cell doesn't know what is right or wrong. The cell is just receiving the signal from the outside world, and the G-coupled protein receptor that it is binding to is relaying that signal in cardiomyocytes. So now we have an overactive heart, we have overactive cardiomyocytes, we have excessive contraction, we have an increase in heart rate. We now have alterations in diastole and systole, we have alterations in blood filling, we have alterations in diastolic volume. So all of these things are impacted because these little proteins that we can't see with the naked eye are losing their regulation. They're not doing what they're programmed to do, right? So we know this pKa pathway is a stimulatory pathway. We know that its signaling tells the heart to increase contractions. And then we started talking about some other things that this pKa does, right? So when pKa is overactive, and I'm just showing arrows here, right? This protein kinase A is overactive. And what does that mean? Well, that means that cyclic AMP is turning on all of the pKa in the cell, and you know that it's being turned on because it's becoming phosphorylated, right? And I'm just going to draw this here like this, right? We know that, oops, that's the wrong color. Let me back up. We know that in order to be turned on, a phosphate has to be attached to one of the amino acid residues, right? So overstimulation of pKa will also induce some changes in the gene. And we talked about this. I told you that this is going to turn on this cyclic AMP response element binding protein. This binding protein, it has been reported it's been reported in some papers as being a binding domain, right? Or a precursor to a binding domain. So basically it tells DNA synthase where to come to start transcribing and making new mRNA. Well, one of the proteins that pKa is going to push into the cell through this cyclic AMP response element binding protein is a protein called G protein coupled receptor kinase 2. And this is present in the cell in small quantities because it plays an important role in recycling G-coupled protein receptors. So we always have some of this in the cell. It acts as a mechanic when a G-coupled protein receptor starts getting up there in age. Let's say this one here has been in the cell membrane for 3 to 6 months maybe. Well, then it's the job of GRK2 to turn it off for a little bit and recycle it and repurpose it, right? So that's what happens in a healthy heart. But when we have something like chronic heart failure and chronic overstimulation of that alpha subunit pathway, well, then what begins to happen is we start to push more of this GRK into the cardiomyocyte. And GRK is going to do what it's been designed to do, which is to inhibit or temporarily shut off the G-coupled protein receptor or the beta-1 adrenergic receptor. So it's basically saying, okay, receptor, there's just way too much of this epinephrine and norepinephrine that is being secreted. This pathway of this stimulatory pathway from alpha subunit to adenylene cyclase to cyclic AMP to PKA, it's just on too much. So we're going to just kind of stop the signaling for a little bit, and we're going to desensitize it. And that's what GRK2 is there to do. It is there to bind to the subunits that are beta or gamma, or the location in which the beta and gamma subunits once were, so that the receptor cannot be shut off or turned on again. So it's just basically inhibiting it for a short time. So when this receptor, when GRK interacts with the receptor, what it's going to do is it's going to put some phosphates on, let me change the color. It's going to put some phosphates on the C terminus of the receptor where the beta and gamma subunits were. I did read 2 papers that said very different things. It said GRK2 will bind where beta and gamma subunits once were. And I read another review where it said that it will bind to beta and gamma subunits and inhibit further mobility. Which one is the truth? I don't know. I'm just telling you both sides of the story as they are presented in pretty complicated research. So they're going to put some phosphates on the C terminus and shut this thing down for a little bit. And they'll find certain residues. Maybe they'll use cysteine. Phosphates love to be placed on cysteine residues. And if you don't know what a cysteine is, that's an amino acid. Of course, this receptor is a protein made of thousands of amino acids, and it's an amino acid sequence. So there's probably hundreds of cysteine residues in there, but this GRK2 protein, it will target certain residues on the tail, right? So even if epinephrine and norepinephrine are still binding, GRK2 is interrupting the signal, all right? So let's move on. Okay, so I took that picture that we just kind of looked at, right? This should be familiar to you. I just went through that and animated it. And I zoomed it out and I put a new receptor next to it, right? So that makes sense, I hope. So here we have a G-coupled protein receptor. It's overactive. It's continuously stimulating that alpha subunit GTP movement to adenylene cyclase to cyclic AMP, right? So now let's talk about the physiology and how this gets shut down so that you can see it, right? Because the papers aren't going to give you the best visualization of this. You'll just be reading words and be like, I'm so confused. I don't know what's happening here. So I told you we have GRK2 in the cell. It is an enzyme that's always present, but with overstimulation, such as with chronic heart failure, we are going to get over-transcription and translation and an increase in that protein in the cell. We call that upregulation. So the GRK2 gets upregulated. Thank you, PKA. We appreciate you doing that. And I told you that one of the things it's going to do is it is going to add phosphates to the end of the G-coupled protein receptor on the tail end, and it's going to look like this. Where is that animation at? Here it is. So GRK2 was recruited, it bound to the tail, it had given up some phosphates, and it had shut down that receptor. So now those phosphates are blocking the beta and gamma subunits from coming back, right? Because the beta and gamma subunits have been reported to be able to return and reset that receptor, right? So now that those phosphates are there, it's blocking anything from reattaching. So the coupled part of the G-coupled protein receptor is now unable to dock back at the G-coupled protein C terminus, right? So this is temporarily off. It has been desensitized. Now, what these phosphates do is they will recruit another protein that will be involved in this kind of evaluation of the protein or of the receptor. So we are going to see another protein that is going to sense those, let me turn this off here. It is going to, sense those phosphates, and it is going to be recruited, that's what we call it, to bind to the G-coupled protein receptor. And it is going to use those phosphates as a tracking system and a binding system. So this protein called beta-arrestin, right? It's arresting, it makes sense. It's arresting the activity of the G-coupled protein receptor. This is going to play a role in removing the receptor from the membrane. Now again, this occurs every day in normal physiology. Beta-arrestin and GRK2 are not our enemies. They are there to keep you alive and keep you safe, because if a receptor starts getting old and doesn't do its job, it is GRK2 and beta-arrestin's job to remove that receptor and put a new one in, right? So these are not bad guys. They are just overactive when we have that overstimulation from epinephrine and norepinephrine. So it's a dose response, right? If we get a higher amount of epinephrine and norepinephrine binding to the receptor, well, we're going to get a higher concentration of SERC and beta-arrestin in a same dose manner, right? So the longer this signal's on, the more the DNA will push out GRK2, and it will probably start upregulating more beta-arrestin so that it could take care of more receptors that are faltering simultaneously. So one of the things that beta-arrestin and GRK is going to do is it's going to desensitize the receptor to stop all signaling, and then it is going to evaluate what that receptor is doing or should be doing. So this will make this complex that will essentially cause something called internalization. And internalization is when this receptor will be taken back into the cell, right? It's just like if your car breaks, you got to take your car to the garage. They bring the car into the garage and then they evaluate it, right? Same thing is happening to the receptor, but we are using a mechanism called AP2 and clathrin. You don't need to know what those are. It's not very important. I just want you to understand the big picture. So essentially, the way this receptor gets inside of the cell after GRK and beta-arrestin is through this vesicle that is made of AP2 proteins and clathrin. This makes clathrin-coated sheets, which makes a vesicle. Again, you don't need to understand it. Just understand that the receptor is attached to this vesicle and it is internalized, right? We endocytose, right? It is taken inside. And this is where the internalization occurs. Now, once the receptor is internalized, we essentially will have two fates. In normal physiology, the fate is commonly the machinery in the cell will review the receptor. It will make sure there's no genetic mistakes. It is making sure that the receptor is acting the right way. Again, use your analogy of a car. When you take your car to the garage, the first thing you do is plug in their computer to your computer and they run an analysis, right? They're just trying to see what lights are going on, what the computer is telling them is wrong with the car. This vesicle that is being endocytosed and internalized is going to do the exact same thing. and the receptor will have two fates. The first fate, if everything looks right with the receptor, the endosomes will basically remove the phosphates and the receptor will be put back into the membrane, right? That's what happens in normal physiology. There are times too when those receptors are degraded and you just need to put new receptors in, right? Because everything has a shelf life in our body. So the phosphates are removed in the endosome. If you don't know what the endosome is, it's okay. You don't need to know what it is at this point. Here is that spelling. If you're like, I've never heard of that. This is the endosome. It's just, it is a mechanism in the cell that will help repair things and help evaluate things. It's an organelle. And that's all you really need to know at this point. Now, the second thing that happens more commonly in chronic heart failure is the endosome is bypassed and the receptors are destroyed. And this happens within something called the lysosome. And the lysosome has all of these enzymes in there, which are hydrolytic and will basically destroy proteins. And if the receptor is sent to the lysosome via the clathrin-coated vesicle, well, then the receptor is destroyed and it is not put back in the membrane. So this is one of the characteristics, one of the key characteristics of chronic heart failure. So while there are all these adaptations that are occurring to try to increase the heart rate, and that's coming from the sympathetic nervous system, there is this series of activation pathways in the cardiomyocyte, which is ultimately leading to the destruction of the receptors. So it's funny, you're playing tug of war here with two different signals. You're playing tug of war with one signal, which is saying, hey, let's increase contraction from the sympathetic overflow. And then you have the cell saying, okay, we got that, but if we do this too much, we're going to activate these kind of death pathways, right? And then you have the cell kind of perishing. So it's a tug of war between let's increase contraction and let's destroy the cell. And in chronic heart failure, this is more than likely the path that is going to be overactive as we move further onto that continuum. So we have this degradation of, you can see here, the receptors being chopped up inside of that lysosome, and it's not being put back into the membrane. So one of the things that's occurring with chronic heart failure is not only are we getting these signals that are increasing contraction, increasing firing, right, from the alpha subunit pathway, right, and then because of that we have issues with ventricle and atrium refilling. But we're also activating these other pathways which are destroying the ability for the cell to contract, right? So these two signals are working against each other. And what this looks like now, if I come over here, what this looks like, I just kind of created a pathway for you here, right? So we have chronic heart failure that leads to sympathetic overflow, right? We have this increase in sympathetic output. We have these changes in cell behavior because we're activating this alpha subunit GTP stimulatory pathway. And over time, that's activating PKA, which is then going to increase the expression of GRK2. And what I mean by the expression of it is the DNA is going to make more of it. GRK2 is going to increase. The more GRK2 we have, with more of these receptors being activated because of the sympathetic overflow, the more beta-arrestin that is going to be recruited, bind to the receptors. And again, on your cardiomyocytes, you might have thousands of these receptors, so you got to desensitize them all at one time because of the erratic behavior of the cell, because of the sympathetic overflow. This is going to lead to a process of increased internalization. And then ultimately in chronic heart failure, this is going to lead to an increase in receptor death. And this is why they call it chronic heart failure, because the heart is eventually going to fail because the receptor, which is the key mediator between the signal, telling the signal secreted from the nervous system, right, central command, that signal is being ignored by the receptor, and the heart cells or the cardiomyocytes can't follow the instructions because the receptors are being removed and destroyed, which is why they call it chronic heart failure. Eventually, those heart cells, those cardiomyocytes, are going to fail because they can't communicate sufficiently with the outside world, because that mediator, which was the G-coupled protein, is now being destroyed. So this is the alpha stimulatory pathway, and I want to go into one more just to show you how the beta gamma pathway also induces this failure, this failing of the cardiomyocytes. All right, so now let's look at this other pathway that has been proposed throughout different research journals. So we talked a lot about the alpha subunit stimulatory pathway, and we talked about how the alpha subunit requires this GTP energy, and it will activate cyclic AMP, and its primary target is PKA. I say primary target because there are thousands of targets in the cell that cyclic AMP works with, but we don't have time for that. We are just focusing on the predominant pathway. So now the other pathway that is activated is the beta gamma subunit pathway. And there is speculation that this pathway is opposite of what we see in the alpha subunit pathway. And we know the net effect of the alpha subunit was calcium control, contractile control, transcription of translation of that GRK2, right? And we could even assume, I bet if we looked in the literature, we would see that this overactivity of this pathway also increases transcription and translation of that beta-arrestin. And what does transcription and translation mean? Just in case you're new to this, it means that the DNA is going to push more of that protein into the cell. So the targets now of the beta and gamma subunits have been proposed to be something that all of you should be familiar with, because this is also true in skeletal muscle. We have these same proteins in striated skeletal muscle as we do in the cardiomyocytes. And this pathway has been proposed to focus on PI3K, which is an enzyme, AKT, and mTOR. And you should know these proteins because we talk a lot about them in muscle synthesis and muscle growth and increases in strength due to muscle resynthesis, because this pathway is activated. Now, interestingly, in skeletal muscle, it takes insulin to activate the PI3-AKT-mTOR pathway, as it is an anabolic pathway. But in the skeleton or in cardiomyocytes, this pathway is being activated by G-coupled protein receptors and the sympathetic nervous system. So slightly different caveats there. Now, one of the things I really wanted to show you here is that because this receptor has two pathways, they do different things, but in chronic heart failure, they kind of converge onto the same sort of thing, which makes this even worse. Remember, I told you with the disease, it's always like a domino effect. One thing goes wrong, and then everything starts to go wrong. And that's how a disease will spread faster or exacerbate and get worse. Well, in a similar fashion, this signaling pathway will also impact gene expression or push more proteins into the cell in a very similar way. So when we have overstimulation of this pathway, AKT, which is a very, very popular pathway in skeletal muscle signaling, will communicate with a transcription factor called NF-kappa B. And a transcription factor has the ability to move from the outside of the nucleus into the nucleus. As you can see here, I have that arrow that's showing you that AKT will add a phosphate to NF-kappa B. It'll turn it on. NF-kappa B will translocate into the nucleus. It will bind to certain promoter regions within the DNA. And then what will it push out? It will push out more GRK2. And then we know the story with GRK2. That GRK2 will be, it will increase its expression. And now it's increasing its expression because we're getting this from the alpha subunit and the beta subunit. And now we have two different signals that are increasing desensitization of the G-coupled protein receptors. I drew an arrow with PI3K as well because there has been speculation that PI3K also independently upregulates GRK2, but the evidence, I didn't have enough hard evidence. I'm just suggesting it right now. Okay, so basically what we have here is we have an increase in GRK2 production because we have two different pathways that are capable of activating the genes to produce more of it to push it into the cell. So now there's another problem that happens with GRK2. As we talked about, cyclic AMP needs ATP, right? We need a lot of ATP in the cell, and that's why I kind of told you the mitochondria health is super important. Well, it appears that GRK2 possesses the ability to cause mitochondria destruction. So when we have surges in GRK2, it does interact with the mitochondria within the cardiomyocytes, and this is going to inhibit the ability for the mitochondria to create more ATP, which is going to inhibit the ability for ATP to get converted to cyclic AMP, which is going to impact this pathway, which is this calcium control, contractile control pathway. And if there's no gas in the tank for contraction, we aren't going to have contraction, and then you're going to have failure. So not only is the ATP necessary for cyclic AMP production, but we also know that with the alpha stimulatory pathway, if we have a contractile control pathway and we want the heart to increase contraction, if there's no ATP, it's not going to be able to bind to the myosin heads and create contraction. So this is everything. This is showing you how in chronic heart failure, and this is one pathway, but we're introducing the disease to you. We're showing you how all of these things go wrong and eventually create complications on a continuum and then lead to receptor destruction, and then we start to get mitochondria destruction, and then one by one these cardiomyocytes begin to perish. They're no longer functioning, and their receptors have been desensitized. So this is all I really have to share with you on the chronic heart failure, and we're going to start moving into myocardial infarction when I return. So please look at this over and over again. Watch this as much as you can. Draw it, pause it, draw it. Make sure you understand all of these things. And with these proteins right here, if you don't know what they are, it's okay. Just be familiar with them. Just know that PI3K is a protein, and I put this on that cheat sheet I gave you. Know that AKT is a protein. Know that mTOR is a protein. Know that NF-kappa B is also a protein. So all these proteins, they interact with one another. They just exchange phosphates, and they turn each other on or they turn each other off. And then ultimately, the reason we signal from the top, which is here, all the way to the bottom, is because we want to communicate with the genes, right? So that's why all this is happening. The end of the pathway is gene expression or gene inhibition. And if the pathway is pathogenic, well, the gene response or the DNA's response is also going to be pathogenic. So good luck with this. I'll see you back in class. Have a good day.